Hello to folks as you're joining. We'll be getting started in just a minute here. Looking forward to a great event coming up. All right. Well, we are a couple minutes past the hour. We've got a full agenda for the day, so let's get started. Haley, if you could just start recording and the live stream, please. Perfect. All right. So welcome everyone. Hello, and so glad that you're able to join us today for the first COVID Info Commons webinar of 2021. Happy New Year. Hope many good things ahead. And we've got many great speakers for you today. As a reminder, this is a monthly webinar organized by the COVID Information Commons project team that is brought to you by the Big Data Innovations Hub with many thanks for support from the NSF Convergence Accelerator. So we've got a really great lineup today that we're happy to share with you. At the end of the event as well, we are going to be talking a little bit more about the COVID Information Commons Undergraduate Student Paper Challenge. This was announced at the end of last year in our December event, and it's an opportunity this spring for undergraduate students to participate in ongoing COVID research and learn more about all of the great work that scientists such as those about to speak with us today are doing. I've included a bit.ly here to the student challenge, and our student Helen Yang will be discussing this further later on in the event. In the meantime, however, I'm truly delighted to be able to introduce you to our 7 speakers today. We've brought together these researchers who are funded by COVID awards from NSF on a variety of different topics from all around the nation. And they're here to present brief lightning talks on this work and hopefully to help build some of the connections that we've been really excited about here at the COVID Information Commons. It's a very networked community. It's a very collaborative community. And we do encourage you to reach out to speakers and others who are doing work similar to your own and see if there are opportunities for collaboration there. Before we begin, I'll just remind you, please share your questions via the chat window. Our student Macy will be moderating this and a Q&A will be held at the end of the event today. Our speakers will also be keeping an eye on the chat. I encourage them to follow up if there are any questions that you're able to answer directly in the chat. And any that we don't catch will be saved for the Q&A portion at the very end. So without further ado, oops, going back too far. There we go. So without further ado, I'm really happy to introduce our first speaker today, David Mendonça of Rensselaer Polytechnic Institute, who will be presenting on his eager. And I will stop sharing so that David can pull up his slides. David, we are ready when you are.
Will do. Thanks, Katie. Happy to do this, and first want to thank NSF for the support, and also to acknowledge the many colleagues working on this project with me. What I want to talk about today is really the role of engineered structures and services in supporting basic life safety, especially food and shelter during the response to this pandemic, and also to consider some of the implications of our initial observations for research and education, particularly in the fields represented by the people you see here. The first example I want to consider is really the food supply chain. And this is a critical infrastructure that is in a state of near collapse. And I don't think that's a dramatization of the situation. We're looking at a near-term shortfall of about 8 billion meals in the United States, as well as a drop in the number of volunteers and individual donations that normally come in. I think one of the real problems here is that we actually do have the supply, but because the demand on the other end isn't sufficient to pull that supply through, we're not getting enough food to enough people. Grassroots organizations and non-governmental organizations are trying desperately to adapt their way of working to meet this demand, but it's extremely difficult to do so. I think our main takeaway here is that we've really got to conclude that some of the assumptions about the resiliency that's supposed to characterize this system could potentially be very, very wrong. Second, again, sort of moving away from food and now to where people live. Lots of people do live in prisons. About 2.2 million Americans live in prisons these days. Prisons have really proven to be the petri dishes of COVID transmission. Around 20% of the prison population in the US is thought to be infected at the moment. One of the approaches that's been proposed is a selective release of nonviolent prisoners, and that's been done to some extent, but really minimally. And one thing that we can't do with prisons is sort of redesign them for social distancing. So this is also an issue. In parallel, there's sort of eerie parallels here to the case of nursing homes. Again, about the same number of people in nursing homes and related facilities, 2.2 million, limited visitation, limited movement, and as with prisons, outsiders are really the vector for infection. It's worth noting, and this is sort of where the pandemic joint hazard element comes in, is that the risk mitigation guidance for COVID is really social distancing along with other measures, but this is directly at odds with what the risk mitigation guidance is for things like hurricanes, which we saw recently with Laura, which is congregation. We're going to see more of this and more sort of disagreements between risk mitigation policies as we go forward. Pandemics aren't going anywhere. I think with both of these settings, we really have to ask, where are the opportunities for the residents of these facilities to adapt their behaviors in order to minimize risks to themselves, and what kinds of tools are we providing for that? I think it's probably no exaggeration to say that we are in the midst of probably the longest and most extensive reverse evacuation and shelter-in-place operation in human history. But instead of leaving home for other spaces, we've really left other spaces for home, and we really have no choice but to stay there. As a result, if you lose your home, you lose not just a place to live, but potentially a place to work and to learn. So viewed from this perspective, private homes are now critical. We need them in order to do the business of the country, learning and everything, but they're not supported as other so-called critical infrastructures are. Overall, I think what our work is bringing out is sort of two main sets of relationships, and one is the first one between adaptation and criticality. As society adapts to hazards, we find ourselves reinventing our notions of what's critical, and vice versa. And as we consider this relationship, we've got to consider the extent to which our infrastructures, our built environment relates in realistic terms with regulatory frameworks. I want to conclude by saying that we feel that there's an urgent need not just to support research that will address these concerns, but really in the longer term, we maybe need to develop our own adaptive capacity as scholars and educators in order to learn from the errors that we've seen in the past and respond to current challenges and shape the design of the future. Thank you.
Thank you very much, David. Really fascinating and critical work being done. Moving on to our next speaker, as a reminder, you are welcome to put your questions for any of our speakers in the chat at any point, and we will hold a Q&A later in the presentation. So Mauricio, please feel free to go ahead whenever you're ready.
Can you see my presentation now, the presentation mode?
Yes, thank you so much. And this is Mauricio Terrones of Penn State.
Yeah, thank you very much. So I will talk about what we are currently doing, trying to develop a portable device for the rapid identification of viruses, including COVID-19, using optical spectroscopy, which would take you, let's say, less than 5 minutes in detecting. So we're using, in our case, we're using carbon nanotubes that you can see here, aligned carbon nanotubes. We use them as filters, and we're trapping viruses and enriching samples from patients. And the idea is to then optically detect it with Raman spectroscopy, which is a very fast technique. What you see here, usually when viruses mutate and you have different sort of variants or mutations, is that the envelope protein of the viruses starts changing, and then your antibodies are no longer effective. And that's a problem. Sometimes you need to develop antibodies so that you can identify them, for instance, by PCR. But if you can trap them without the label, without the antibodies, that's what we're trying to do. So we're trapping new viruses and also trying to trap them and concentrate them without the antibodies. We're using aligned carbon nanotubes, so we can grow different arrays of aligned carbon nanotubes, and we can control the intertubular distance of the tubes. So in principle, we can trap viruses of different sizes, because the viruses range in size and morphology as well, depending on the types of viruses. The technology that we're using to concentrate viruses it's this one. So it's a size-tunable enrichment platform. And what you see here is that when you have your sample, you pass it through, it's a microfluidic device. You are trapping your viruses in this three-dimensional filter that later on you can do different things with it and in order to identify. But we are concentrating the viruses very effectively. So here is, you see an SEM image of the viruses. This is H5N2 that we have enriched and we have trapped. And then you have the viruses, some of them adhere very well to the carbon nanotubes. And then we have a technology to enrich viruses. And that's the technology we're currently using to use Raman spectroscopy, which is an optical technique to detect them. So what we do is we make these large devices, I mean, sort of microfluidic devices, which are larger, and we use gold, in this case, gold nanoparticles to get a Raman signal. So you shine a laser, you get a signal, and then you get a spectra. So the type of spectra that you are getting, it's, for instance, for different viruses, you get these different peaks, different viruses, different concentrations. So you can use machine learning algorithms and then identify and detect viruses. And this is something that we're trying to do right now. So we have done it with flu viruses, and these are the current flu viruses that are circulating as well in the flu season. And you have your vaccines, flu vaccines included the H1N1, the H3N2, and the flu B. And what we have seen is that you can see the different signals from the different viruses, that you get the signals from the envelope proteins, and we can identify with accuracies larger than 95% different types and subtypes of viruses, which is very good. So the project that we are currently doing with the IGER is really moving forward as well in this direction, because we're trying to do coronaviruses, and we're using coronaviruses from chicken. And here we are trapped. You can see the trapped viruses of the coronaviruses from chicken. So this is just comparing the respiratory viruses from humans and the chicken coronavirus. You can see is the same range, 5 to 2,000, 500 to 2,000, 500 to 2,000, and you can see more peaks here. So you have different peaks coming from the coronaviruses. So one of the things is, can we differentiate using this technique, differentiate different strains of viruses? And we have tried 32 strains of these viruses. This is the chicken coronaviruses. And we can see that depending on the year and the region, we can really identify them. So you can see they are grouping them in different sort of areas. That means that we can really, in principle, we can identify different strains. And that's very, very important for our work. In addition, we have also identified this receptor binding domain from the spike protein of SARS-CoV-2, the COVID-19, and we can see the peaks and the signal from this domain. So right now, what we're trying to do is we want to improve the signal to noise ratio. So we want to get better, even better signals using 2D metal, so using other substrates instead of the gold nanoparticles. And we're getting these sort of sharper signals. So we get sharper signals and better signals. Then we will be able to have even higher accuracy and better performance. And we want to deploy this in the field. And for doing that, we have this collaboration with JPL, with Sona Hosseini. She can make this sort of handheld Raman spectrometers, which you can deploy in the field. So that's what we are currently doing. We hope to finish this design, and we hope to get an improved signal to noise ratio over the following months. And this is the team of researchers. These are the students and postdocs involved, and Sona Hosseini as well, who is part of this award. Thank you.
Thank you very much, Mauricio, for this great talk about all the great work you and your team have been doing. Next up in our lineup, we have Tracy Van Holt of New York University, who will be discussing the pandemic's impacts on the U.S. food supply chain. So Tracy, we're ready whenever you're ready to share your slides.
Okay, just get to the, perfect. Everybody can see everything?
Yes.
Yes, okay, great. So we're back to some of the food supply issues. First, I just want to thank our team, project team across New York University, University of Florida, and Arizona State, and National Science Foundation for this work. So we're a team of geographers, cultural anthropologists, and supply chain experts. So of course, we've all seen the news. When the pandemic hit, there was news reports of discarded products, and part of this really highlighted a couple of things about our food supply chain. One, this is a picture of tomatoes, and tomatoes in the US basically are for the restaurant supply networks. Tomatoes for supermarkets essentially come from Mexico, and this is according to my colleague who works with tomato farmers down in Florida. And so our supply chain was really designed for efficiency in the restaurant and supermarket network. And what we're seeing as all the restaurants are closing, that the system is collapsing in many ways. So, restaurants now, of course, don't have as many customers, but they're still struggling to get products at the same quality and price. And they've also had to adjust their business models, as we know, for social distancing, et cetera. So this work is really looking at the pivotal role that restaurant supply networks really play in the food system. We're focusing on independent restaurants. They're really important in our economy. Small businesses, there are 500,000 small businesses that are small restaurants. 11 million people are directly employed, and 5 million people in the supply chain. And a lot of small scale farms are also supplying these restaurants. And they're facing different sets of operational challenges than larger franchises and larger businesses. So this is why we focused on them. So our focus is really to understand the vulnerabilities in the supply chain, as well as in restaurants and how they are adapting and responding. So a couple of questions we're addressing is, what are the strategies that restaurants are using to adapt? And does their relationship with not only their suppliers, but other restaurants help them? Because there is very much a restaurant community. And finally, how has the supply chain shifted? So we're working across our 3 sites. One is in New York, in Queens, and in Manhattan. We're also focused on Maricopa County and Gainesville, Florida. So as the pandemic has unfolded, of course, COVID, which was once a hotspot in New York, is now a hotspot in some of the other places. So it really works for a rich research. Some of our methodological advances in supply chain research is one, it's really, there's not a lot of focus on spatial analysis for adding that component, where we've designed our research that's Zoom friendly, but based on ethnographies. And finally, social networks. Even though supply chains are networks, there's a paucity of information on supply networks. And so specifically, the vertical networks, the supply chain, as well as the horizontal networks, the restaurants. So this is, one of our contributions. I'm just going to show from some of our semi-structured interviews with the restaurant owners. At the beginning, we've coded for these interviews, and basically I'm going to show you a visualization of the themes. And so what you're looking at here, don't be afraid by this pretty figure here, are all of the words that restaurant owners were talking about. And we see two really big divisions. On the right-hand side, there's really focused on supply chain issues, distribution, relationships with suppliers, alcohol sales, drivers taking other routes, competition with other chains. And then on the left-hand side, these are really operational issues. So it's, how is the restaurant pivoting? How are they finding, keeping their staff safe with mask wearing? How are they, a lot of their employees have left. So how are they handling this? And finally, just on the bottom, you could see a lot of this pivoting to takeout. French fries don't travel well, as we all know. So there's a lot of food that have been designed for restaurants in one way that now that they've had to pivot, has an effect on everything else. If we just focus on the main themes in all of these, it really boils down to a couple of topics in our interviews we're conducting now. We're focusing on this. Again, this is like zooming into the most prominent themes. On the right-hand side, it's really about distribution and planning and the supply chains. Suppliers are not providing products at the same intervals as they were in the past, even if they are available. And then really on the left-hand side, the COVID risk to staff is a big issue for the restaurant owners, getting their clients to wear masks when they're not eating. And then we see this sort of intermediary in terms of pivoting their business model and pivoting to takeout. So just in conclusion, there's a lot of factors that are contributing to restaurant outcomes, and we hope that this research will inform not only information on strengthening some of our components of the supply chain, but also develop resilience to the future. And finally, if you want to reach out to us, either me or any of our team members, feel free to do so. And to learn more, you can see our website. Thanks so much.
Thank you very much, Tracy, for a really informative talk. I encourage, especially if we do see some of these conversations starting in the chat that I am seeing now, I encourage speakers, if they feel like leaving their contact info in the chat for people to access, please feel free. We'll also be adding some additional links and other resources there later on today. So being mindful of time, we will move on to our next speaker, Michel Boufadel of New Jersey Institute of Technology. So Michel, we are ready when you are.
Okay, yes, sure. Just,
Perfect.
Okay, do you see the first slide?
We do, thank you.
Okay, so this is the project Rapid. My name is Michel Boufadel, I'm the PI, and Dr. Xiaolong Liu Gang is co-PI. We collaborated with various researchers in the nation. You can see the list here at Princeton, Duke, Rutgers, Hopkins, University of Pittsburgh, and the University of Cincinnati. The first part of the talk is going to focus on the number of cases in the US. So we obtain these data from Johns Hopkins University, and and they provided them daily. And then so we analyzed the spatial distribution of the number of cases. And then you can see here in March 2020, and then in May. And then if you zoom in on a certain region, let's say this is the Washington, DC area, we noticed like the number of observed cases, they are spiky. So you have like the high number, maybe this is DC or Baltimore, and then you move, and then there's nothing in between due to smaller population, and then you have a higher number. So for us, that's reminiscent of what's observed in turbulence. And then so we thought, okay, that the number of cases would be something known as multifractal. And so we got to investigate that. So the conclusion is that the number of COVID-19 cases is what you call scaling, and then, but it's not totally random. It is correlated, and we analyze the correlation using what you call the Fourier spectrum. So first, because it is scaling, so you can find a direct relation between what's happening at 10 kilometers up to 2,600 kilometers. So initially, during the early phase of the disease, the correlation was small, as one could deduce from the slope. As the slope gets, steeper, then it means the correlation increases. And then we noticed that the spatial correlation of the disease converged towards the spatial correlation of the population. And these are other multifractal properties that, they are in the paper. I'm not going to discuss them now. And then for our investigation, we used a relatively simple model developed, more than 120 years ago. It is called SIR model, susceptible. These are the people who could be infected, the infected ones, infectious, and then removed. These could be removed to they recover or by death. So we use this model to try to capture what is happening. And if you recall that spectral slope figure, this is here shown as a time series using also our model, which is the line. And then one could note that we were able to reproduce the spatial correlation using that model. This is just an illustration of how our model functions. So we start with the population that is multifractal, and then we assign the model for infection, and then you can see these are the number of new cases. Of course, with time, the number of new cases would subsides. The conclusion of this is that the first thing was the major finding for us was the spatial distribution of population is multifractal. So which allows us to explain why the COVID-19 spatial distribution is multifractal. There are major work where they use big data to model the spread of the disease using number of people, using their phones. So our approach, provides a compromise between the big approach, the big data approach, and fitting models at small towns, say at the scale, say of Newark. And there's always issues of privacy using big data. And the other, again, this is maybe pure modeling, but we believe that paying attention to spatial correlation would constrain the model so it doesn't go wild. The next part of my talk is about the movement of viron, or it's called them particles, in the supermarket. Imagine this is a supermarket that is 40 meters long, 20, 25 meter wide, and then you have the doors here, the red arrows. These are the where the air comes from, vents, and then the white arrows are the return vents. This is hypothetical. And so we use CFD simulation, the Trans, to model the movement of air in the supermarket. And I want to show here the results. Our focus was on the attachment of the particles. There are a lot of studies they deal with the transport. But for us, we say, okay, what happened, because we do know that the virus or the particles, they do attach to surfaces. So here you can see them attaching to the ceiling, the orange, they attach to shelves, blue attached on the floor, which is yellow. Whereas if you don't allow for attachment, even after 20 minutes, you see them spread all over the place. So therefore, the attachment on surfaces is important when you want to predict the indoor transport of viruses. This is one curve here where you have one graph. You have the concentration at 5 meters from a source, the source. This is without attachment of 5 micron droplets, so it is 20% the strength of the source. with 25% attachment, you can see this is like maybe 12%. And then with 100% attachment is like 10%. So we conclude that the attachment doesn't play a role, which means the type of surfaces in the supermarket is not going to play a major role, because there were discussions like, oh, should we use metal or glass or plastic? Based on these simulations, it seems it doesn't make a big difference. One thing we investigated is also, when they said, okay, there's one way in the supermarket so people could walk one-way aisles. And then we said, okay, well, one of the things that you could reduce, the number of the virus particles in the air is maybe you can create baffles. This is, as an environmental engineer, we're used to using to this concept for plug flow reactors. And then we conclude that if you place these baffles in the system, you are going to reduce the concentration of particles in the air. And the other thing that from the study is that the narrower the aisles, the better the air quality, which is kind of counterintuitive, because whenever you look at supermarket, you look at large aisles, and then it gives you the feeling that it is healthier. Thank you.
Thank you very much, Michel. Definitely something I'll be thinking of as I pick up my groceries, but super relevant and interesting research. Our next speaker coming up is Jamie Hestekin of the University of Arkansas. Jamie, we're ready whenever you're ready to share your screen.
Can you see my first slide?
We sure can.
Okay.
You can just go into presenter mode. There you go.
Yeah, thank you very much for this opportunity. Well, this is a rapid project started in June, and I am the PI, and the co-PIs are Dr. Krista Hestekin and Dr. Peter Crooks. So what I wanted to tell you all about, first a little bit about, is long-term disinfectants. So this project is to make long-term disinfectants for disinfecting hard surfaces, soft surfaces, lots of different surfaces. We were working on an NSF project where we had, we're making tempo modified cellulose, so tempo oxidized cellulose, and we found that by attaching certain groups, we could get it to be a disinfectant. There is currently only one long-term disinfectant that has been approved for emergency use by the US EPA. There's the website dealing with that. Normally it takes about 18 months to get a disinfectant approved. This one was obviously approved faster because of COVID. There is a need for long-term disinfectants for both bacteria and viruses for now and when COVID beyond. We're working with the University of Arkansas Medical School on these long-term disinfectants. And what we're really looking at is all of what goes into this. What's the best chemistry? What's the best application procedures? And what sort of potential lifetimes can you expect from these surfaces? And this started as part of this NSF App score, as I said. So, and if my slide will advance, then we can, okay. So we started looking, we are making a lot of these different chemistries. I'm going to show you one specific result today, but the easiest way to make the chemistry is ionically bonded materials. There is long lists of approved disinfectants for different viruses and bacteria, and you can take those disinfectants and ionically bond them to the surface, your own surface, and so then coat that surface onto whatever hard surface you want. So that's the easiest form. That's the form over here on the left. One of the more difficult forms is we have something that will make a peracid. And so what this will do is it will make hydrogen peroxide over the course of time. And so by doing that for long term, you can put an oxidizing agent in which will destroy things. So that's a material we're testing as well. And then the third one is to try to take these chemistries and covalently attach them to the tempo modified surface. We've developed the ability to put click chemistry on the surface of these tempo groups. And so because of that, we can really attach just about anything we want. And we're looking at some of the approved disinfectants for doing that. What this shows you is some of our initial results. So this is with the easiest form, the ionically bound disinfectant. We used a common disinfectant, DDAC, and attached it to form 1, which is one form of our tempo modified cellulose. The form 1 says that every other carboxyl or every other hydroxide free group has been converted to a carboxyl, and form 2 is one where they're all converted, and so it becomes water soluble in things. And what we did here is we used E. coli as a first test. This is how fast E. coli grows when you look at it on a 96 well plate. And these are the different things that and how they're growing out. And so if you look at this, our form 1 doesn't do much at all to stop the growth. The DDAC and the form 1 with the DDAC both stop the growth when you did that. You move on to 48 hours, you can see that our surface is doing pretty well. After a week, after 2 weeks, we're still seeing close to complete, basically a complete ability to destroy E. coli when we put it into the surface. And this has a lot of error bars because these are all 96 wells apiece that are doing this. And the reason I put it on here, it gets kind of messy when you do all that stuff. But over a 2-week time period, so far, we can see that these ionically bond surfaces work pretty well. As far as how they work on the surface, I'm going to start a video here. As you can see, and I guess on this video, that we have a surface that we've coated this on with a spray sort of surface. It is a green spray surface because we put the colorant in. We can scrape it, but if you just rub hard on it, it does not come off. If you spray it with water and a little bit of surfactant, on the other hand, you can get it to come off very easily. And eventually here in about 5 seconds, it's going to show that you can remove it all and your door handle looks the same way that you had before. Right now we were adding, the green color has nothing to do with science. It's a marketing thing that people are interested that how would you know that something had this spray done, but you can see it can go on and off pretty quickly. And so right now we are also tests, which I'm not going to show today, of rubbing hard on this surface to see, you know, how much you can get off of the surface and such. And then just the last really quick slide, because I know my time is coming up, is we started with bacteria because bacteria is easier to test, not necessarily easier to kill, but viruses are a little bit difficult. We are in the process of doing that right now to show we'll do that as well. We are developing the covalently bond groups. And there's a lot of interest in this sort of thing, and we're interested in collaborating with any one of you as well. Thanks. Bye.
Thanks so much, Jamie. Really appreciate that talk and the demo there. Our next speaker today will be Wai-Yim Ching of the University of Missouri, Kansas City. So Wai-Yim, you are welcome to share your screen and proceed whenever you're ready.
Okay, can you hear me?
Yes, we can.
So I have can you see the screen?
Not yet.
Okay.
Have you hit the share screen?
I should, but I'm not in the. Sorry for the.
That's okay.
Share the screen. Yes. Share the screen. I'm sorry. Can you see it?
Not yet. Oh, there you are. Great.
Okay. I'm sorry for the following up. So, so I think let me go to the first slide. I restrict myself only to 5 slides, and I'm Wai-Yim Ching from University of Missouri, Kansas City. And I'm a theorist, computational theorist. And so my result would be very condensed. And I think anybody is interested could send me an email. I will answer all the questions. So we have a very small team and consists of the Hikari and 2 very capable graduate students, PhD student, Mr. Jawa and Sen, and an undergraduate student, Ms. Namik. So we are supported by NSF, a LAPD project, which is in the condensed matter and materials theory section of NSF. And since we are a computational scientist, we have to rely a huge amount of computational resources. And this was provided by the Department of Energy Supercomputer Center and called NSF. So I want to start the next slide. And so I say something about the background and why we want to start to study the spark protein, because this is the protein binds to the host cell receptor. Most of the people know what is going on there. And so it plays a critical role in the infection. So the structure and the properties of this and internal bonding is not fully understood. So we have to use the world-class supercomputer facilities to do ab initio calculation based on density functional theory for this large molecular system. And this type of calculation is very rare, and because it's very resource consuming, and it's very challenging. So our goal is to have fundamental understanding based on quantum mechanical methods and interatomic and intraatomic amino acid network, including the hydrogen bonding. So we also, of course, the main focus is to train the next generation of scientists in this very important area of material science, because I was trained as a material research, and I work in condensed matter physics, in chemistry, and biophysics. Okay, so biomaterials are very important. So I want to first report some of the latest achievement, and we have the structure optimization of 7 basic structure domains in the spark protein, which is right here. This is the free chains, and the chain A is has 7 different domains, and each of them consists up to several thousand atoms, and we have to do the computation for all these 7 domains together, and investigate their covalent and hydrogen bonding and the partial charge distributions. And we also developed a key parameter called amino acid bond pairs, which would be able for us to do the 3-dimensional amino acid interactions. So we so far have published 3 papers. If any of you are interested, please send me an email. So currently the ongoing project we did is the interface modeling, which we combine the molecular dynamics with DFT calculation, and this is even more challenging. And we also try to calculate the rigidity of each of the spark domains, because the rigidity is very important in any biological system when they are under temperature change or stress. We are also currently working on the mutation modeling, which is the D614G, which if is in the news, that those are the variants of different coronavirus, and it's in the news almost every day. We also want to improve the computational methods and the codes used for large data generation. So this is a figure about the the mutation model with water molecules included. And this is the preliminary result on the rigidity. And so we have a free manuscript currently now under population that will take us at least 2 or 3 months. You know. So this comes down to the one of the last slides. So our project will finish by the May 31st, which is only a 1-year project for the NSF Rapid. So we would, before we end the project, we will extend our computational modeling to drug design, but for some selected models by adding of the very short peptides. So this is an example of two models which we would sort of start to model, which is called LCB1 and LVB3. These are the two short peptides to be inserted at the interface. We also will do extensive mutation modeling and on the analysis of the following cases. And in addition to the one we are currently working on, there are many new variants according to the B117 report from COG United Kingdom. You know, so these are the 4 examples, and there are many more. You know. And we also want to, longer goal is to extend computational modeling to cover much larger biomolecular systems. Even though we are doing our initial calculation at this moment, it's one of the largest computation that can be envisioned, but we are more ambitious, want to extend to even larger systems. So on the other hand, we are very ambitious. We want to advocate the use of large-scale computational modeling for biomolecular systems that can rival or even complement the experimental techniques in accuracy and at a much reduced cost. Thank you very much. Yeah, that's the end of my talk.
Thank you very much, Wai-Yim. Really appreciate your insights here. So for our last researcher today, I'm going to introduce Rabindra Tirouvanziam of Emory University. And it looks like we have your slides up here. So please feel free to go ahead whenever you're ready. All right, if you are speaking, we can't hear you.
Let me make sure I'm unmuted. I was just saying thank you for the opportunity, and it's nice to be closing the session after such a good series of talks. I'm an immunologist and an engineer here at Emory University in Atlanta, and the work that I'm going to be talking about was funded by an Eager Award by CBET. And it was done really by a grad student in my group, Brian Dobosh. A lot of the bioinformatics done by a postdoc in my group, Diego Moncada. And all the BSL-3 work, really the work with the virus itself, was done in collaboration with Kevan Zandi and in the laboratory of Raman, who's a prominent virologist and pharmacologist here in our university. So as you guys know, SARS-CoV-2 is infecting a number of cells in the body, throughout the body, is causing that disease COVID-19, which is really a multi-organ disease. And even though I think we need to think about the different organs that are being affected, most of the morbidity and mortality in patients is linked to the lung manifestation, and this is really due to the infection of the cells lining the lung, the epithelial cells, and that leads to in the complications of the disease, the influx of monocytes and neutrophils from blood, and then these cells sort of compound on the initial issue that was caused by the infection of the epithelium. So our mandate really was to use a system that we developed for other diseases in which we grow human airway cells at air liquid interface so that they mimicry the conditions in which they grow in the lung, and we can infect them with the virus for different periods of time, And then we can let different sets of immune cells, primary immune cells from human blood, transmigrate and meet the virus on the other side. So we're really trying to mimic the sequence of events. So the viral infection, then the monocytes coming in, then the neutrophils. We can add drugs at any point in time to affect infection or immune response. And then we can obviously analyze the different components in that model. And we use a lot of different omics methods. Our goal is really to characterize the steps in pathogenesis and also to check for potential benefits of candidate drugs. And I'm going to show you a few data that basically report on our progress. On the epithelial side of things, obviously you want to make sure that in our system, the virus behaves similarly to what we've been seeing in vivo. This is really the case. I'm showing here a quick comparison of SARS-CoV-2 with PR8, which is an H1N1 influenza A virus, and OC43 that you can see here is one of the common cold coronaviruses. And right away, you can see by RNA sequencing and heat maps that I'm showing here of a few families of genes that, with the influenza virus, antiviral genes are really hot, so they're red, but the coronaviruses are really much colder. So this is one of the areas of interest in pathology and also in therapeutics. Coronaviruses, and especially SARS-CoV-2, seem to be very good at preventing the activation of antiviral pathways in epithelial cells. On the other hand, if you look at the cytokine genes here, there's really discrepancies between the three viruses. And we can see right away that SARS-CoV-2 is able to activate an IL-10 response, but not so much an IL-8 response. So it's really promoting more of a monocytic inflammation to begin with, and neutrophilic inflammation comes after. So this is really what we see in vivo. Now, the model is really unique in its ability to combine epithelial cells and virus and immune cells. So we can, for example, look at in the process of this infection of those two cell types, we can look at the effect of drugs. So baricitinib is an immunomodulator that was approved by FDA. Remdesivir is an antiviral, and they work through different mechanisms of action. There's obviously interest in combining them. So we can see that we can combine both drugs, can actually block the migration of monocytes secondary to the infection of the epithelium. So that might be responsible for some of the benefits that we see for those drugs when they're combined in vivo. We can also look at the viral burden in the different compartments of our model, in the epithelial cells, in the monocytes, in the extracellular fluid, and we can combine all of those to look at the total viral burden. And you can see again that the combination of remdesivir and baricitinib in 6 to 10 different replicates here shows a decrease in the total viral burden. More importantly, we can also look very much in depth at the molecular response in terms of transcription and all other functions that you're interested in the epithelial cells, in the leukocytes. In this case, I want to illustrate our ability to show, for example, in the context of no drug, that the monocytes that are infected by SARS-CoV-2 show a huge decrease in the transcription of interferon and also the STING RNA sensor. But by the same token, the infection is increasing the transcription of IL-1 beta and also IL-8, which we know are going to lead to the recruitment of neutrophils. And again, we can look at the effect of single drugs or drug combinations in that system at the transcriptional level. What's really interesting is that there was a paper in Nature yesterday showing that alveolar macrophages show a huge burden of SARS-CoV-2. At the time that we were working on our study, we actually didn't have a lot of data going in that direction. So we realized by computational means, this is work that we did in collaboration with the Gosen lab at Emory, some single-cell RNA-seq data from bronchoalveolar lavage of mild and severe COVID patients who were all hospitalized. And we found a population of monocytes in the lung that show exactly the same type of transcriptional activation upon encounter with the virus, where they are very high for IL-8 and IL-1 beta. So we think our model mirrors in vivo data, both on the epithelial side, also on the leukocyte side. So in summary, we have the ability to recruit monocytes, infect them by SARS-CoV-2. They produce a pro-neutrophilic response. All of this is mirrored between the in vitro model and in vivo situation. there are a number of questions, obviously, that we're asking in this model, both in terms of the virology, the immunology. We have now this model under provisional patent for drug testing, because we think this is sort of the most urgent effort that needs to be put together. And we're testing a number of immunomodulatory, antiviral, and pro-repair drugs. More in the next few months on this. I'm just going to finish here by acknowledging folks in my lab, folks in the Shinazi lab, Gosen lab, Gibson lab at Georgia Tech, who helped us with some of the transcriptional analysis. And again, the EAGER award that was instrumental in having us start this project and pivot from our work in lung immunology to COVID-19 research. And this is my contact. I'm going to stop there, and I'm happy to take questions in the chat. Thank you all.
Thank you very much, Rabindra. Really appreciate that in-depth talk. And again, I encourage people to share their questions in the chat. And speakers, please feel free to drop your email addresses in there to help facilitate any sort of back and forth that may emerge from this. So to be mindful of time, I'm going to go straight to our student presenter next. We'll save some of the remaining questions for the end if folks are able to stay on a little bit past the hour. I know we're running a bit long. But I wanted to make sure that we got to the undergraduate student paper challenge I mentioned at the beginning of the hour. So our next speaker will be Helen Yang, a student at the Northeast Big Data Innovation Hub, who is going to tell you, I know we have a few students on the line today, fill in a little bit more about the challenge, including some of the resources that are available for undergraduates to leverage. So Helen, please feel free to go ahead.
Thank you so much, Katie. Hi, everyone. My name is Helen Yang. I'm a student at Columbia University and an assistant at the Northeast Big Data Innovation Hub. So we announced the launch of the COVID Information Commons Student Paper Challenge during last month's webinar, and we've been thrilled to see so many students and researchers already interested in participating. So today, we're excited to announce a few important updates and share some resources that students can use to develop their paper ideas. So for a quick overview, the KIC Student Paper Challenge is an opportunity for undergraduate students of all academic backgrounds around the world to learn about COVID research and apply what they're learning in class to current real-world issues. So the webpage for the challenge can be accessed from the homepage of the KIC website at covidinfocommons.net. The page contains all the information students or researchers interested in serving as mentors or judges need to know. So essentially, the challenge for students is to leverage the KIC resources to write a 4 to 5 page double-spaced paper on a topic related to COVID research of their choice. So as an exciting update, which we've coordinated with the Columbia University Libraries team, is that winning papers will have the opportunity to be published not only on the KIC website, but also added to the Columbia Academic Commons, an archive of scholarly work produced by Columbia students and affiliates. And authors of winning papers will have the opportunity to present their work at a future KIC community webinar like this one. So the webpage lists key dates for the challenge, a few of which have been updated since last month's launch. Students interested in participating should submit the participation form, as well as a title and short abstract for their paper by February 15th. We're also looking for mentors interested in holding office hours in late January, February, March to help students develop their papers and judges to help evaluate the submissions. So if you're interested in sharing your COVID research experience with students, please fill out the mentor/judge participation form by March 1st if you're interested in serving as a mentor, and March 15th if you're interested in being a judge. Filling out the participation forms and checking back to this webpage will ensure that you'll be kept updated with any new announcements about the challenge. So we recently recorded a video going over all of the information about the challenge and that this webpage provides. We also recorded a video demonstrating how students can leverage some of the resources on the KIC website to help develop their papers. You can access both videos on the challenge webpage. To give a brief summary, we invite students to explore the 3 search tools that KIC provides for the over 900 COVID-related research projects the NSF has funded to date. These include the COVID Research Explorer, a tool that uses machine learning to visualize research trends, the NSF Awards and PI database, our most recently developed resource, or the simple NSF search by directorate. There are a variety of ways students can use these tools to learn about research projects and find a topic they'd like to write about. For example, the COVID Research Explorer tool provides tree map and topological map views that group awards based on common keywords and other fields that students can customize. Students can adjust the settings of the maps to analyze how research has changed over the course of the pandemic or differed according to region, for example. And like researchers, many of you who have been using this tool to identify collaboration opportunities, students can also look at potential cross-disciplinary areas of research. And in the query box to the left, students can add search terms to learn about research on different topics. The COVID Research Explorer tool provides details about individual awards, but another resource students can use to learn about specific projects is the KIC NSF Awards and PI database, which can be accessed from the homepage or the menu of the KIC website under KIC Search Tools. Here, students can easily search by keyword and filter their results. For example, typing supply chain into the search bar brings up 28 results. Clicking into any of these awards, again, provides full details about the project. A unique feature about this database is that clicking on the name of the PI will take you to an individual page with some information about the PI and their work. Many PIs have provided additional information about their projects, such as their lab websites or articles about their progress. Students can use this information as a starting point to learn about specific projects. And finally, for a general overview of COVID-related research, students can look at projects being funded under different NSF directorates, which we've linked on the main page of the KIC website. Students can explore different projects under a directorate related to their major, for example, to get a scope of the research being conducted in the discipline. So there are many more resources besides these three tools available on the KIC website that students can use, including curated data sets, guides, and articles. Students are also welcome to use resources not on the KIC website. So we'll wrap up this overview for now, but for a more in-depth demonstration of how students can use these three KIC search tools and leverage other resources, please watch the demonstration video at the bottom of the challenge webpage. And if you have any questions about the challenge, please email info@covidinfocommons.net. We hope you're as excited as we are about this opportunity, and please share it with any students who might be interested in learning about and joining the discourse on COVID research, and consider participating yourself as a mentor or a judge. Thank you so much.
Thank you so much, Helen, for that great overview. I know that we already have a number of students and several mentors and judges who have signed up to participate, which is really exciting. And we do encourage any of the folks on the call today, if you're interested in serving as a mentor or judge, please do sign up. And for anyone who has any questions, please feel free to share this in the chat. Helen, if you could actually share the bit.ly for the direct link to the page in the chat so that folks could get a closer look if they'd like to.
Yes,
that
absolutely.
would be cool. I think we had a few questions from our fantastic speakers earlier as well. So Macy, if you could cover any of the questions, if you could read any of the questions that aren't yet answered from the chat, please.
Yes, thank you, Katie, and thanks again to the speakers for the presentations and for keeping up with questions in the chat. We have one remaining question, which is to Wai-Yim, and Miranda Lynch asks, have you explored how to add glycans to the structures for the spike protein being used in the computations, since it is known to be heavily glycosylated?
Yes, so this question is addressed to me. I'm in the process of modeling many different types of scenarios that other researchers are interested in. And so I want to establish any possible collaborations with their suggestions. But since my knowledge in the biology is very limited, so I would ask people to send me directly by email, so I can have a lot more detailed planning or discussion to you. I'm sorry about that.
Thank you, Wai-Yim. Are there any additional questions, Macy, at this time?
No, that's it for questions in the chat. Thank you.
Awesome. I do see a remark from Anastasia Angelopoulos, apologies if I've mispronounced that, about the abstract submission deadline. Thank you. We really want to consider how best we can encourage students to participate. I'm going to just drop our contact email in the chat. Please feel free to reach out to us. And if there's any way that I think we can help your students participate, I'm sure we're more than happy to discuss that. And I see Florence has added a remark there as well about the timing that we've pulled together for this challenge. So, well, I know that we're a bit over the hour and I want to be mindful of time. I do encourage folks to reach out to the speakers and for these conversations to continue. We've had many exciting collaborations occur over the course of these events, and it's so excited to see them launched again in the new year. Our next event will be coming up on February 10th. And I will just pull that screen up real quick so that you guys have that. But it is February 10th, 2021. And we'll have another lineup of 7 more great speakers. And we do encourage you to register and join in. So thank you again to our speakers today for their excellent presentations. Thank you to NSF and the entire KIC project team for all of their support. And many thanks to our great student moderators who have chipped in today. So we'll leave you with that. Thank you.